Introduction. UV absorption spectrometry is a widely used analytical technique that utilizes the absorption of UV light by molecules to provide information about their electronic structure and concentration. Next we discuss working of UV absorption. Number 1, light source. The instrument is equipped with a UV light source that emit a broad spectrum of UV light, typically in the range of 190 to 400 nanometer. Common light sources include deuterium lamps and xenon lamps. Number 2, monochromator. The UV light passes through a monochromator, which consists of a prism or diffraction grating. It disperses the light into a component wavelengths and selects a specific wavelength of analysis. The monochromator also includes adjustable splits that control the width of the light beam, ensuring a narrow and well-defined wavelength band. Sample holder. The sample to be analyzed is placed in a transparent cells which is made of quartz or other suitable material that transmit UV light. Measurement and detection. The cuvette containing sample is inserted into the spectrophotometer sample holder. Number 2, the UV light beam passes through the sample and sample of light is absorbed by the molecules present in the sample while the rest is transmitted. Data analysis. The spectrophotometer measures the absorbance of the sample at the selected wavelength. A calibration curve or a standard solution with known concentration are used to relate the absorbance to the concentration of the analyte. Now we discuss application. Number 1, quantitative. It is used in quantitative analysis, drug analysis, environmental monitoring, biochemical analysis, and industrial applications. What is uses of UV absorption? Number 1, structure elucidation, kinetic studies, drug development, forensic analysis. Number 1, structure elucidation. The UV absorption spectrum of a compound provides information about its electronic transition, which can aid in the determination of its structure and bonding pattern. Number 2, kinetic studies. UV absorption spectrophotometer allows monitoring the rate of chemical reactions by measuring the change in absorbance over time. This information is essential for studying reaction mechanism and determine reaction kinetics. Number 3, drug development. The UV absorption spectrophotometry utilized in the early stage of drug development to determine the absorption and pharmacokinetic properties of potential drug candidates. And last, forensic analysis. UV absorption spectrophotometry is used in forensic science laboratories to analyze trace evidence, detect drugs or poisons, and identify unknown substances in the crime investigations. Thank you so much.